Okay, so welcome back to this next video in which we are discussing the different types of lipid molecules. Okay, so we've just discussed the structure of a triglyceride or a triacyl glycerol molecule, sometimes also called a neutral fat molecule. Okay, and what we now want to discuss is the structure of phosphoglycerolipids. And if I say phospholipid, the structure that you will probably think of is what's more correctly called a phosphoglycerolipid. Okay, because there are other forms of phospholipids. Okay, all of the phosphoglycerolipids are phospholipids, but not all the phospholipids are phosphoglycerolipids. Okay, right, so we firstly need to discuss something about the optical isomerism of glycerol. Okay, so basically, when you just have a glycerol molecule, okay, and I'll start drawing its skeletal structure because that will save me time. Okay, so in skeletal formulae, remember, you don't show carbon atoms. Carbon atoms are implicitly shown by corners, and you don't show hydrogen atoms coming off carbon atoms. Wherever there's a missing bond coming off a carbon atom, you just assume that that's to a hydrogen atom. Okay, so here are my free carbons. Now I need to show the alcohol groups coming off them. Okay, so here we go. Each of them now has an alcohol group coming off it. Okay, basically when you just have a glycerol molecule like this, you don't need to worry about optical isomerism. Okay, none of these carbons are chiral centers. So for instance, if we look at this one here, this has got the alcohol group coming off it, this C group here coming off it, and then two hydrogens coming off it. So it doesn't have four different groups, so it's not a chiral center. This one is identical to this one, so that's not a chiral center either. The only question is whether this one here is a chiral center, but actually it's not, because it has an alcohol group, a hydrogen coming off it, and then it's got these two groups, but those two groups are identical, so it's not a chiral center. So glycerol only has one optical isomer. The problem comes when you start adding things onto these alcohol groups, because if I add a group, let's say, onto this alcohol group here, and don't add it onto this one, then that suddenly makes these two groups that come off this central carbon non-equivalent, and that suddenly makes this central carbon an optical, a chiral center basically, and means that this molecule will now have two different optical isomers, okay, which will actually be enantiomers of them, each other, which means there will be mirror images of one another. Okay, so in phosphoglycerolipids, what we're basically going to have is a phosphate group coming off one of these alcohol groups here. Okay, so let's say it's going to be this one. Okay, and that is going to break the nice glycerol molecule, which doesn't have any optical isomers, and it is going to result in optical isomers. So I want to now discuss with you the two different optical isomers of that molecule and which one is actually found within cells, so which one's going to be important for us. Okay, so let me now revert back to drawing molecular formulae. Okay, so I'm going to put this carbon, the central carbon here, at the center of my page. Okay, now it's got an alcohol group coming off it, and it's got a hydrogen coming off it. I'm going to have these two coming out of the page towards us. Okay, so here is an alcohol group coming out of the page towards us. This carbon also has a hydrogen atom coming out of the page towards us. The other two groups, which are these two carbons, they're going into the page away from us. Okay, so I'm going to have this carbon over here, which we'll call the first carbon, up here. Okay, so that's just a CH2OH group. And then the carbon that's going into the page away from us down here, here this is, it'll have two hydrogens coming off it. Okay, it's then got this alcohol group, but actually I'm going to attach onto this alcohol group a phosphate group. So I should just actually discuss with you how you attach phosphate groups to alcohol groups. So let me show you the structure of a phosphate group. Okay, so a phosphate group consists of a phosphorus atom at the center with a double bond to an oxygen atom and 2 alcohol groups coming off, and then a single bond to another oxygen, which has then acquired electrons via ionic means, well, an electron via an ionic mean, and therefore has a single negative charge, like so. So this is a phosphate group. Now, basically you can attach phosphate groups to alcohol groups in a very similar way to the way that you can attach carboxylic acid groups to alcohol groups. If in fact you look closely at the structure of this phosphate group here, okay, look at this phosphorus atom with the alcohol group coming off and the double bond to an oxygen. If I replace that phosphorus atom with a carbon atom, it would look exactly like a carboxylic acid group. Forget the fact that it's got 5 bonds, which is totally unrealistic for carbon. If we just look at this group here, it would look exactly like a carboxylic acid group. And in fact, this can react to alcohol groups in exactly the same way as a carboxylic acid group would. So, basically what you can do is you can remove that alcohol group from the phosphate group, remove the hydrogen from the alcohol group, bind those two things together to make water, okay? And then bind the phosphorus atom of the phosphate group to the oxygen atom of the alcohol group. And this link, which is very similar to an ester link but isn't an ester link, is called a phosphoester link. Okay, so this is a phosphoester link. And you combine this phosphate group to the alcohol group via a condensation reaction which yields water here. Okay, right, so that's how you can attach phosphate groups to alcohol groups, okay, via these phosphoester links. And these are very similar to the ester links that we've seen between carboxylic acid molecules and alcohol molecules. Okay, what's different about linking phosphate groups to alcohol groups that carboxylic acid groups don't have is that actually, if you think about this, once it actually has attached onto that alcohol group of the alcohol, then you've still actually got this group over here. You've still got a phosphorus atom doubly bound to an oxygen with an alcohol group coming off here. That group is identical to this group here. Okay, so it can actually link via another phosphoester link to another alcohol group. Now that's not going to be important initially, but that is going to be important later. Okay, right, so of this oxygen here, which is the alcohol group of the 3rd carbon of the glycerol molecule, as we're going to view it as, okay, we are now attaching this oxygen to a phosphate group. So let me show this here, like so. So here is the phosphorus atom doubly bound to an oxygen with an alcohol group coming off it, and then a single bond to an oxygen which has then acquired an electron via ionic means. Okay, so basically you can see that this carbon is now a chiral center. Okay, if I swapped this alcohol group with this hydrogen, so I brought the hydrogen onto this side and the alcohol group onto that side, then that would be a different optical isomer. No matter how you rotate the molecule, or no matter how you rotate yourself and look from a different angle, you will never be able to see this molecule equivalent to the one that I'm just about to draw. So I'll draw out the other one here. Okay, so, oh dear, what am I doing? I'm about to draw the exact same thing. Get rid of that oxygen. Right, I'm now drawing the mirror image, okay, of this molecule here. So if you put a mirror down here, I am just going to take the mirror image of it. Okay, so here's the carbon, okay, the 2nd carbon of the glycerol molecule, and then coming out of the page towards us, we've got another alcohol group here, okay, and then going into the page away from us, here is this CH2OH group. Okay, so the 1st carbon up here, and then also going into the page away from us, we've got the 3rd carbon, which is down here, like so. And I should have just discussed the fact that when I draw a bond like this, where it's fully coloured in, and it's getting thicker towards the end, that means coming out of the page towards us. When I draw a dashed one like this means going into the page away from you. Okay. And then off this alcohol group, you then have the phosphate group down here. Okay, like so. So, no matter how you change the way you look at this, you can't turn it into looking like this one, basically. These are two separate molecules. No matter how I rotate this around, I cannot turn this into this without cutting this bond, and swapping them back over. Okay, so these are optical isomers. Now, when the optical isomerism is so nice that one optical isomer is actually the mirror image of the other optical isomer, then there is a fancy word for that. The two molecules are called enantiomers. Okay, so in this case, we do actually have two enantiomers here. Okay, right. So, what is the naming system for these two different enantiomers? So, basically, firstly, I should just tell you the naming system for the different carbons. So this carbon here is called the first carbon, this carbon here is called the second carbon, and the one with the phosphate group attached to it is called the third carbon. Okay, the same here. So, basically, this molecule here is called L-glycerol, and then it's free phosphate. So this is L-glycerol free phosphate. This one over here, meanwhile, is D-glycerol free phosphate. So this one here is D-glycerol free phosphate. Okay, so that's the naming system for these two different enantiomers. So now what I need to tell you is which one is actually present within cells. It is L-glycerol free phosphate, which is the more important one as far as biochemistry is concerned. It's the one that we find within cells, so we don't care about D-glycerol free phosphate. So, the molecules out of which our phosphoglycerol lipids are going to be made will be these L-glycerol free phosphate molecules. Okay, so that's just a little bit of a warning with regards to these different optical isomers. We're not going to really stress on this later on when we come to discuss the phosphoglycerolipids. Okay, right, so let's now discuss what a phosphoglycerolipid actually is then. So basically, the phosphoglycerolipids is a large family of molecules that are all based on a type of molecule called a phosphatidate molecule. And just like there was not just one triacyl glycerol molecule, there is not just one phosphatidate molecule. It's a whole class of molecules that would all be considered phosphatidate molecules. Now, phosphatidate is strictly speaking the conjugate base of a molecule that you could call a phosphatidic acid molecule, so a phosphatidic acid molecule. Okay, so either phosphatidate or phosphatidic acid. Okay, right, so let me show you now the structure of a phosphatidic acid molecule. I'm sorry, I can't really say that. Phosphatidic acid. Okay, right. So, let me now show you this. So, basically what we're going to take is we're going to take an L-glycerol free phosphate molecule and we are going to esterify long-chain carboxylic acids to the alcohol groups that come off the first and the second carbons of the glycerol molecule. Okay, so we're going to put long-chain carboxylic acids off these first two, and then off the third one we will have a phosphate group. That sort of a molecule is called a phosphatidate molecule. Now clearly there is not just one phosphatidate molecule, because you don't have to have a set long-chain carboxylic acid esterified to each of these alcohol groups coming off the first and the second carbons of the glycerol molecule. So there is a scope for variation basically there. Okay, right. So basically this is what you do. Okay, so we'll start with the glycerol molecule. Okay, so here are the first two carbons of the glycerol molecule. Here is the third carbon, and we know that the third carbon has attached to its alcohol group a phosphate group, like so. And I'll discuss what the difference between phosphatidate and phosphatidic acid are in a moment. Okay, but basically it's on the level of this phosphate group here. Okay, now what we're going to do is esterify to the two alcohol groups coming off the first and second carbons of this glycerol molecule, long-chain carboxylic acids. Okay, so here is the carbonyl group, and I'll then put an R here to indicate that it's arbitrary, basically. And similarly, in the second position, here's the carbonyl group, and I'll put R prime. So these two groups don't need to be the same, basically. You can have one long-chain carboxylic acid here and a different long-chain carboxylic acid here. So once again, I'll stress that there is not just one type of phosphatidic acid molecule. There are many different molecules that would all be classified as being a phosphatidic acid molecule. Okay, right, so now to discuss what the difference between phosphatidic acid and phosphatidate is. So basically, strictly speaking, what I have drawn here is phosphatidic acid, okay? If this proton on this oxygen here was to fall off, okay, and go into the free solution, leaving this oxygen here with a negative charge, that would then be the conjugate base of phosphatidic acid, and that's called phosphatidate. Okay, so strictly speaking, this is the conjugate base. So remember, an acid molecule is a molecule which can donate protons into a solution. Okay, so this molecule that I've drawn here is an acid molecule, hence it being called phosphatidic acid. Okay, because it can donate this proton off this alcohol group here, and therefore it's an acid. Okay, however, once it has donated the proton off, what is left is no longer an acid molecule because it can't donate any more protons. It doesn't have any more to donate. Okay, instead it's actually a base because you've got this oxygen here with a negative charge, and also this oxygen here with a negative charge, which are both essentially equivalent after they're both just oxygens with negative charges, and they can accept protons back from free solution. So protons that are in the free solution can come out of solution by binding to these negatively charged oxygen molecules on the phosphate group of a phosphatidate molecule. Okay, now things which accept protons are called bases. So phosphatidate molecules are actually bases. Now this is a general principle for all acids, that once they've given away their protons, what is left is then a base molecule, and it's then called the conjugate base of that acid. So phosphatidate is just the conjugate base for phosphatidic acid. Okay, right, this is the molecule after which all of the phosphoglycerolipids are going to be derived from, basically. Okay, they're all going to be based on this molecule. Now, this, if you like, is the core glycerophospholipid. Okay, so what are the other phosphoglycerolipids that you can make from phosphatidic acid? Okay, well basically what you can do is you can add different groups onto this alcohol group that comes off the phosphorus atom of the phosphatidic acid molecule. Okay, and by adding different molecules on here, you will end up with slightly different phosphoglycerolipids. Okay, so let's talk about some of these different phosphoglycerolipids that you can create then. So firstly, let's start off with phosphatidyl choline. Okay, so all of these phosphoglycerolipids, they have phosphatidyl as their prefix, and basically what this means is that you start with the phosphatidic acid molecule, and then you're going to stick another group, okay, in this case choline, onto the phosphatidic acid molecule, and that will create you the final phosphatidyl choline molecule. So let me show you the structure of a choline molecule. So basically, choline is an alcohol molecule, okay, and it's a 2-carbon alcohol molecule. So here is our ethylene group, and we'll have an alcohol group at one end, and then at the other end you have something a little bit more interesting. You have a nitrogen atom, which has then got 3 methyl groups coming off. Now this is the same choline as is present within acetylcholine, the famous neurotransmitter. Okay, in acetylcholine you just bind an acetic acid molecule via an ester link to this alcohol group here, and that's acetylcholine. Okay, so this isn't acetylcholine, this is just the choline molecule. Okay, but this is the same thing that is a component of acetylcholine. Okay, so you remember me talking about how we could use a phosphate group like this to actually form two phosphoester links. We formed one here, but even after we form that, there is this other group here left over, and we can use this again to form another phosphoester link with alcohol groups. Here is an alcohol group, so we can link this phosphorus atom here of this phosphate group of the phosphatidic acid molecule to this oxygen atom of the alcohol group of choline, and that then will create us a phosphatidyl choline molecule. So basically, you will take this alcohol group off here, you will take this hydrogen off this alcohol group here, you will bind that phosphorus atom to the oxygen and bind the alcohol group to the hydrogen to create water. That then creates you phosphatidyl choline. Okay, right, let's now look at another phosphoglycerolipid. Okay, so the next one I want to talk about is called phosphatidyl ethanol amine. Okay, so phosphatidyl, so once again we start off with a phosphatidic acid molecule, and then we're sticking off the phosphatidic acid molecule an ethanol amine molecule. Okay, so again we've got an alcohol group here. Okay, and I should just say that phosphatidyl choline is usually just abbreviated to PC. Okay, right. So let's now turn our attention to phosphatidyl ethanol amine. Well, we know not to be afraid of the phosphatidyl. That just tells us that we've got a phosphatidic acid molecule. The ethanol amine, that's again really simple. Okay, so basically this is just a simpler version of choline. Okay, so let's draw our ethylene group with an alcohol group coming off it again. So here's the ethanol. Okay, and now we've got an amine group coming off here. Okay, this is just simpler than choline because it's just got two hydrogens rather than three methyl groups. And I should have said, in the case of choline, the nitrogen here has got a positive charge because you have three methyl groups coming off it. So nitrogen shouldn't have 4 bonds like so. Okay, it should only have 3 bonds ideally, and then 1 lone pair of electrons. And the fact that nitrogen has 4 bonds means that in one of these covalent bonds, nitrogen has put both of the electrons that are within the covalent bond. So if you remember, between two molecules, well, between two atoms, a covalent bond should consist of 1 electron from one atom and 1 electron from the other. Basically, what will have happened is nitrogen has donated both electrons into one of these bonds, okay? And when it does that, it's effectively donating 1 electron away to the other member of the bond, in this case a carbon, because the usual understanding of a covalent bond is that 1 electron came from each of the members. So basically, when you've got 2 electrons sitting here, it's as though one of them belongs to the carbon and one belongs to the nitrogen. So effectively, when the nitrogen put both electrons in, it gave one away to the carbon, and that's why it got a positive charge. So you might ask, well, surely that means that the carbon should have a negative charge. Well, the answer to that is that the carbon will come along initially with a positive charge, okay? And when it received this electron from the nitrogen, it then became neutral, and it deferred the positive charge off, if you like, onto the nitrogen. Okay, so that means that the overall choline molecule is positively charged. So this is the ethanol amine molecule, and then you can guess exactly how we're going to attach this onto our phosphatidic acid molecule. Basically, we're just going to make another phosphoester link. We're going to take the alcohol group off this phosphorus atom here. We're going to take the hydrogen off this alcohol group here. We'll bind the two together to make water, and then we're going to bind this phosphorus atom here to this oxygen atom here to create another phosphoester link. Okay, so that's phosphatidyl ethanol amine, and it's another example of a glycerophospholipid. Okay, right. Next up, I want to talk about Phosphatidyl Glycerol. Okay, right. So, Phosphatidyl Glycerol. And basically, this time what we're going to do is attach that phosphate group of the phosphatidic acid molecule onto another glycerol molecule, basically. Okay, so let me show you this. So, basically, if we draw out our glycerol molecule again, so here's our glycerol molecule, it's got these 3 carbons, like so, and we know that each of these carbons has an alcohol group coming off, like so. Okay, and basically what we're going to do is attach the alcohol group of this first carbon over here onto the phosphate group of the phosphatidic acid molecule. So again, we're just going to form a phosphoester link. We'll take the alcohol group of the phosphate group, we'll take the hydrogen off the alcohol group of the glycerol molecule, and then we'll bind the oxygen of the alcohol group on the glycerol molecule to the phosphorus atom on the phosphatidic acid molecule, and that will give us phosphatidyl glycerol. Okay, right. Now, if you are concerned with the fact that this carbon will be a chiral center, then you are quite right, it will be a chiral center. So let me now tell you the optical isomerism of this carbon here. So basically it will look like so. Here is the oxygen here of this alcohol group here of the first carbon that will be bound to the phosphatidic acid molecule. Okay, then here we'll have this first carbon, here we'll have the second carbon, and here we'll have the third carbon. Okay, now off the third carbon we'll then have an alcohol group. So let me just colour in these carbons to make sure that you're getting this picture. So here's the first carbon, here is the second carbon in turquoise here, and here is the third carbon in green here. Okay, so we just now need to, I just now need to tell you whether the alcohol group comes out of the page towards us and the hydrogen goes into the page away from us, or whether the hydrogen goes comes out of the page towards us and the alcohol group goes into the page away from us. But I think we'll call it there for this video, and we'll continue this discussion in the next video. So I'll leave that one as a cliffhanger.